There's a big world of difference between running, how do you say it, academic research, where you are using a liquid biopsy as such as your treatment selector, compared to actually establishing a liquid biopsy in the routine clinical practice. It's two different worlds, of course. So moving the actual liquid biopsy assays that we are having today to the clinic requires, of course, a lot of training of departments of molecular diagnostics within hospitals to really how to use this technology properly. It requires a lot of education, a lot of training from that perspective. Also from a reimbursement perspective, the reality is that, for instance, I'm from Belgium, and where we have reimbursement driven medicine, the price tag per diagnostic test is fixed. So there is this, how do you say it, also artificial ceiling above us that actually limits us to implementing a liquid biopsy test that isn't, I mean, to be frank, it is an expensive test. It's true. So if you then also have this bottleneck of the reimbursement, that's something that we need to tackle in the long run as well, by really lobbying with the authorities that really go over these technologies to really assess what the cost efficiency would be to actually implement these types of tests. And then, of course, the availability. The reality is today that the test sets we are having today, these are so-called lab-developed tests in the academic research. There are commercial alternatives out there, but again, implementing them in the real-life practice is not that easy. So if you want to establish, for instance, our very own lab-developed test with the upcoming IVDR regulation, we need to be ensuring that this particular assay is compliant, that we really harbor this, that basically your test as such is compliant to all regulations. Are there other examples of trials that are using a liquid biopsy? Of course. So I'm just thinking about one of the top of my head is, for instance, the AMPLITUDE trial, where we are looking into the use of niraparib plus abiraterone in men with metastatic hormone-sensitive prostate cancer, where patients are also being selected for harboring a DRD or an HRD defect, where they are using either a tissue or a liquid biopsy to really screen the men to assess eligibility to be enrolled in the trial. Another example is the PC-BETS from the Vancouver Prostate Center, where based on the liquid biopsy result, patients are being randomized to different targeted treatment arms. And I flag that there are multiple examples currently where people have embraced the use of using a liquid biopsy test during the screening process of our patients.